Cool. Good afternoon, everyone. I just want to talk about now some of the work I've been doing whilst I've been based at University College London since I moved from sort of doing invertebrate work, which I've spoken about at Palace in previous years. So I sort of regenerated into a sort of pseudo-vertebrate paleontologist, been doing some of this work. But just before I start, I just throw out a few acknowledgements quickly to mainly my colleagues at UCL who've dealt with me ramblings the last couple of years and learning more about vertebrates in general, and the various contributors to the paleobiology database. I don't need to list them all, but I've put a few key names there of some of the main contributors to the data, and also to Richard Butler and Paul Barrett for discussions on some of their data sets, which I'll get to later in the talk, and not least of all, of course, the funding came from Leverhulme. So what we've been interested in is the latitudinal biodiversity gradient through deep time. And many of you will be aware of this as a very well studied but yet slightly poorly understood macroecological pattern. It's represented here by this picture taken by a review by my co-authors in Trends earlier this year. And it just shows the distribution of species on the planet as we see it today through a heat map, and the large red colours obviously represent areas of high biodiversity. You can see the sort of concentration of biodiversity around the equator with a drop off into these sort of colder, bluer colours, north and south respectively. But we're interested in how this pattern occurs through deep time and in different groups, and what potentially these drivers might be, if indeed it shows a similar, or as I will get to, a very different pattern in deep time to what we see today. So in order to do this, we've been using a large data set compiled over several years. As I've said, I mentioned some of the main contributors at the beginning from the paleobiology database. And this represents somewhere in the realm of 19,000 individual occurrences for Cretaceous terrestrial vertebrates across the whole globe. And this represents somewhere in the magnitude of about 2,700 odd species. I don't want to go into too much detail about the second part of the slide because I appreciate I'm probably going to go over, and Emily will kill me. But because a lot of the data in the paleobiology database is either unidentified to a certain taxonomic rank, or are kind of CFs and AFs that may well be associated with different species or considered to be indeed separate, we sort of set up this taxonomic protocol in order to maybe get a bit more data out of the database, especially dealing with things that are in debt, because there's a lot of data in there, and then able to try and clean up the data somewhere. But I don't really want to go into that in too much detail. But we do have a lot of data, really, all in. This is all the data shown on a modern map, with the reptiles in blue at the top and the mammal data in the bottom. But you can see that certainly with the reptile data, we've got decent coverage for most of the globe, although that's kind of largely concentrated in places that you would expect: North America, Asia, and in South America to a great extent. But the mammal data in red at the bottom is obviously shows a lack of data, but more specifically, we're missing a lot. There's a lot more in-depth, unidentified fragments in the southern hemisphere, which we've tried to deal with over the last couple of years, but it's hard, really, essentially. So we don't have as much data for mammals, unfortunately. But it's just to give you an idea of how much data we do in fact have. But really, just to start off with the main results, really, is to look at this diversity. These are plots using a subsampling method known as the shareholder quorum subsampling method, devised by John Alroy. And on the left here, we have the data of the original untreated data set taken like download straight from the paleobiology database, and on the right, the data as modified, given the modifications I sort of went over in the last slide. The individual lines in this don't matter so much. They're just different levels of subsampling or quorum levels. So, for example, the red line here represents a quorum level of 0.1, moving up towards about 0.6, 0.7. But the key things to take away here are twofold. Firstly, that obviously cleaning the data or augmenting it to an extent has had a great effect in our ability to get out higher levels of data. You can see certain trend lines, like, for example, the sort of dark blue line, which is about 0.3 quorum, is not very complete on the left, and it sort of gets a bit more complete on the southern hemisphere specifically. So we're able to get larger quorum levels out, which is something we really want. We don't really want to be sampling only 10% of the data. We want that value to be as high as possible. But the other important thing is that, unlike the first slide when I was showing you this heat map of the world, is that the modern biodiversity gradient is not represented in the Cretaceous in terms of terrestrial vertebrates. The peak in diversity is seen very much in the northern hemisphere, around 45 degrees, in sort of paleo-temperate regions, differing very much from the modern realm. This was identified by my co-author Phil Mannion and others in a paper a few years ago, looking specifically at dinosaurs. But what we've shown here by using the entire data set, or wider data set of terrestrial tetrapods, is that it's not restrictive only to dinosaurs. It's quite a consistent pattern. across the whole group, although largely this data obviously consists of dinosaurs because they're very diverse in this time. I don't have time to cut the data up in as many ways as I would like to, but I'm just going to show you one particular example of how consistent this pattern is looking at physiological difference between endotherms and ectotherms on the left and right, respectively. This is to the exclusion of dinosaurs, just to give you an idea that the pattern is consistent when removing one of the most diverse groups within the data set. But it's also, this high peak is kind of, again, it's present in both a warm-blooded and cold-blooded species. It's quite consistent. It's also quite consistent amongst different terrestrial realms and different clays, but obviously I don't really have time to go through every particular point. One thing we also want to do is to look at different exploratory variables that might well be controlling it. And I've thrown a few up here, and they'll all come up in just a second. So we have various measures of temperature, non-marine area, and a measure of sampling. In this case, it's the amount of tetrapod-bearing or terrestrial tetrapod-bearing formations for the Cretaceous, examining these latitudinally on the y-axis. And these we'll be able to sort of compare and see whether these are a driver of this particular pattern. But in order to study this in great detail, so I realise I'm rambling quite a bit here, we are using GLS, or Generalized Least Squares modelling. And in the past, when we've been examining diversity trends and trying to relate them with environmental or sampling proxies, we tend to do lots of pairwise comparisons. But this method is a maximum likelihood framework and allows us to test combinations of variables. So we have all these exploration variables. As I said before, we have a measure of potentially primary productivity taken from plant data, which I'll get to in a slide or two, compilation by Richard Butler and colleagues. And we also have measures of sampling, as I said, tetrapod-bearing formations and some environmental proxies. But we can combine all these together and see which of a combination of these shows the best fit to our diversity pattern, rather than just comparing them all individually and pairwise for all these different variables. We can try PSD and PDF plus PDF, etc., and try every combination of this. When we do this for a whole of subsampled tetrapod diversity, as I said, using the SQS method, I'm only showing the top 3 models here because to show them all would be dull, frankly. The best method that seemed to be coming out in this originally was this plant diversity, this plant subsampled diversity, and really the important number at the top is the AICC weighted value, which is the likelihood value that we use in order to compare these models. And this seemed to be coming out quite strong. But it wasn't so strong when necessarily you compared different groups. Now the reptile group subsampling there again showed a combination of plant diversity and evenness, which is a measure of sort of environmental sampling within each bin, taken from a measure from Rook et al. A few years ago. But in the mammal diversity, it shows a different pattern. Again, we're getting effectively a sampling measure, this evenness value coming out. I spoke to Richard Butler about this at SVP, and he brought up the point that his data largely, his plant data, comes from occurrences where there are dinosaurs, essentially. So that's where they collected their data. So there's a real independence issue with this data. So I was kind of curious to work with that a little bit and just see really if that was such a true pattern, and whether the plants were just being artificially inflated by the fact that they're occurring at the same places that the dinosaur data has been collected. So the first way to get rid of this was to remove plants from the models. And if we do this and use the same analyses with exactly the same variables minus plant diversity, we get a combination of non-marine area, evenness, and temperature, which may make a little bit more sense. It's a combination of the amount of actual land mass. As I said, there's a sampling measure in there too, and as well as temperature. But the AICC value is not nearly as high. If I flip back here, you see that we're kind of in the sort of 90%, 0.9, 0.75. But it's a bit lower here. The other way of doing it is to see if we get a different result with different plant diversity data sets. So using the data available within the paleobiology database. Again, the plants come out as strong, but again, not to the same high level. Other ways to look at the paleobiology database with just plants that occur from non-dinosaur formations. And again, it seems to be coming out again a similar pattern to previous analyses with a combination of sampling and plants. So they seem to be very much true, but if this data is indeed not really relevant or not good enough for this purpose and removing it, potentially we get a pattern that maybe makes a bit more sense. So we have a combination of the amount of land mass and some kind of environmental parameters, although I admit there are several climatic factors I really want to include into these models, such as precipitation and seasonality and such, which I've been trying to get more recently to improve these models as things that you might naturally expect might be helping to drive these patterns. So that's really the broad scale of the analyses. But if these data sets aren't, if indeed the pattern is not well modeled, or we can't support some of these models. I said the AICC values, these likelihood values, were a lot lower. Potentially that's because the origination of this diversity that we're seeing in the Cretaceous actually extends further back in time. And one way I thought I've been doing this in more detail, and this is admittedly just really preliminary work. This is a tree taken as Roger's recent dinosaur tree from his +1 paper earlier this year. And basically what I've been doing is taking the paleolatitudinal coordinates of all these taxa and then performing an ancestral reconstruction essentially along all these nodes. And I appreciate you can't read all these taxa and all these numbers, but that doesn't really matter for this instance. Sorry, I'm flying through anyway. Maybe I should breathe. So maybe it's a Scottish thing, we're all talking too fast. But so these values, these ancestrally reconstructed values, so I'm interested in plotting them through time. And just to make this sort of easier, we've got the Triassic, the Jurassic, and the Cretaceous here in 3 different blocks. And the ancestral values, not for the tips, just for the nodal values of dinosaurs, just specifically dinosaurs, of course. I'm working to analyse in various other groups just to see how this pattern, how widespread this pattern might be. And as I said, this is very much preliminary, and I've got a lot of work to do on this in terms of subsampling some of this data to check the supported of it. But this peak in diversity, on the right-hand side, we have the northern hemisphere. And if we look in the green areas at the top right here, you can see this peak in diversity that I've been discussing. But actually, this is kind of prevalent further down into the Jurassic, at least. Now, there's a possibility that we have a lot more dinosaurs in this part of the world, so you might expect a lot of ancestors to be reconstructed in this way. But then we kind of see down towards the Triassic, we start to get values at the equator, at least, if not in the southern hemisphere, and there are several dinosaurs found in these sort of areas in South America and such in the Triassic. But it does mean that perhaps the reason why we're getting this high diversity in this particular location, and one of the reasons why we might not be able to model it to quite a high level, is because actually the causal reasons for that diversity peak extend further back in time, and in fact there are potentially other measures and other drivers for that pattern. But I said these last 2 slides are purely there as a way to suggest work that's currently ongoing. So you don't need to look at it too much detail. There's clearly a lot more to be done in this area. But in essence, in probably way under time, just to sum up some of these main points, primarily that this latitudinal biodiversity gradient is very different in deep time. It doesn't have this high peak at the equator, as we see in modern realms, but also that there may well be that driver from primary diversity. The plant data, as I said earlier, may have a strong support, but this data itself is maybe questionable for this purpose, for this kind of analysis. And removing it gives a stronger effect for other environmental and area measures and such, as I said. So there's a bit more work to be done in just checking that data. And I'd be happy to talk to anyone who may have really good, or on a better understanding of some of this data. I would appreciate that. But also more likely that we also may have to look further deep in time to see where exactly these taxa are originating and indeed diversifying and moving into. But that's in essence it. So I'll just thank you for putting up with my ramblings for a few minutes, and thank you very much.